Hey everyone, I just wanted to go through cell cycle regulation and cancer notes with you. So anyone who wanted additional information could have it. I feel like we all have at least some family member or maybe a friend or something, some pretty close connection to someone who has gone through um, or experienced cancer in some way. So it's a good thing to kind of just cover and be aware of and have that knowledge set. So I don't want to shortchange anyone on their learning or understanding of this. And I know our bio interactive click and learn HHMI assignment went through it, but there are still questions that linger around it. So I'm going to just kind of go through a few more with you when we get to our genetics unit and start to talk about transcription, translation, and how genes are passed along to one another. We'll have even more information on like when mutations happen and that sort of thing. I have a couple slides here at the beginning that are review for identifying phases of the cell cycle. So if you want to hit pause and practice identifying as many as you can, go for it. Otherwise, I'm just going to keep on going here and identify a few of them with you. Okay, so interphase is when the cell is spending the majority of its time, or is the phase in which the cell is spending the majority of the time in. You're going to see a solid dot or those nucleoli within the cell. So the majority of them here are interphase. Now, I know we can see a couple dots in there, solid dots, and um, so people always ask, is that cancer or is it not? And just typically what is happening here is we just have a um, large clump of genetic material near each other. So one of them would represent like the nucleolus. You might also start to see some of the spindle fibers forming, and that's where we might see other solid dots, but those would be the spindle fibers. So majority here, like this is a really good example of interphase right here. Okay. Our next phase on the list is, remember, IP-MAT-C? I peed on the mat, see? So interphase, our next one is prophase for the P, and that's where our chromosomes start to condense down and we can start to see um, some organization happening. So this is a really good example of that. There's a couple more over here. This is getting into the prophase where we start to see some spacing and not just a big old blob of genetic material that's no longer chromatin, again, chromosomes. So that's a good prophase. M is next for metaphase. This is a great example of a metaphase where we see the genetic material lined up in the middle of the cell. This one is an example of metaphase, or maybe even the prometaphase, that phase that kind of happens between the two that we just talked about here. We can see them lining up and getting organized, but they're not quite fully in that metaphase um, plate, or sorry, yeah, the metaphase plate, which would be the center imaginary line. Oof. So next comes anaphase, where they start to separate. So this is kind of like an anaphase telophase example. Again, they kind of go hand in hand because once anaphase folds and separates the sister chromatids, then telophase starts to kick in and starts to reform the nuclear envelope around that genetic material. And then cytokinesis is two cells that are brand new. So this would be kind of a telophase cytokinesis connect. We don't see, the two cells here that are completely different from each other. So we can still say that they're separating at that point. So that might be a telophase cytokinesis. Here are some more. And again, you can see, you can pause and look for some again, but the majority here are going to be all interphase, where it's kind of the cell doing its normal thing. Then it starts to organize itself into prophase, right? We start to see some organization of prophase here and here. There's a lot more for us to visualize prophase. Next comes metaphase, where they're lined up in the middle. Not too many metaphase in here. This is getting close. Might be a pro-metaphase happening here. And this is kind of metaphase-y, but it's really hard to see that genetic material in its line, right? Then if we're talking to anaphase, telophase, right? We can see the sister chromatids are being pulled apart. Sister chromatids are being pulled apart over there too. This one has a little bit of that. This one, we start to see the separation of our cells here. These are onion cells that are being split apart. So this is the cell plate that forms. So that's our telophase and cytokinesis connection. Again, you can kind of see a line forming here to separate it out for telophase and cytokinesis. Yeah. So we learned about the cell cycle's control system, kind of like that analogy of gas pedal and brake. And there are built-in checkpoints that are going to stop and regulate the cell and ensure that the cell is in good form before moving forward through the cell cycle. So this one looks a little bit different from what I've drawn in class, but here's our interphase all here. And then just the small sliver is mitosis when the nucleus divides and cytokinesis happens, right? So as we go through, before we reach the end of G1, there is a G1 checkpoint, which is going to check growth and make sure that the cell is doing its normal thing and allowing nutrients in and waste out properly. It's going to check and make sure, there's no genetic damage, that sort of thing. And if there is, it'll hit stop and have some more proteins come on in and try and regulate that cell or fix any damage that went on. And if it's good to go, it'll go green light and we can continue on into the S phase where genetic material is replicated. And more growth happens in the G2 phase. And sometimes these cells will just kind of hit pause right here until it's ready to divide, or the cell nucleus says, hey, it's time for you to divide. But this G2 checkpoint is going to check for proper growth so that it's big enough and ready to be split in half. It's going to check and make sure that DNA was properly synthesized and we have the correct amount of each chromosome or chromatin at this point, that to make sure that there is the correct amount of each of them present. So that's that one. And then midway through mitosis, when the nucleus is dividing, this is more along when it hits metaphase, anaphase, and the genetic material starts to separate. That's where big problems can happen. So that's where a checkpoint occurs to make sure, did the sister chromatids separate properly, and do we have the right amount of genetic material on either side of our cell? And then we'll end up with 2 in the end. So those are our checkpoints: go, no go. And we have this G0 phase where our cells kind of get into this resting phase, or it's called the quiescent phase, where they just kind of pause. And if it's at a checkpoint, or if the cell is not ready for it to go through and divide, it might push a cell into this phase where it's just going to rest and hang out until things get solved by our checkpoints, or the cell is communicated that it needs to continue through the cell cycle and go through and split in half. So again, we can see the next 2 go-ahead signals for pro-metaphase, and then again with anaphase and metaphase like I was referencing earlier. So what are they doing at that M phase? They're checking to make sure spindle fibers are attached properly here, and the kinetochore is linked in at the centromere. The sister chromatids are lined up properly and they're next to their actual sister and not a stranger. But they're matched up with their identical sister there. Okay. So again, our control system goes through and just makes sure that everything is okay. I kind of just zip through that, but I'll attach the slides later because this was clearly laid out in the click and learn assignment. So how do the checkpoints get regulated? Proteins are going to do that. They're called cyclins and they're called CDKs, which are cyclin dependent kinases. So the cyclins are going to be a protein molecule that require a CDK to come and attach itself to it. And when they attach, those are going to go through and control that rate and say, hey, we're going to stop for a second, or when they release, they'll continue on. So if you are a graphic type person, we can see concentration amount, right? So as it spikes up, we know that they are present. As it drops down, that means there are less of them. There are different types of cyclins associated to each of the checkpoints, which makes sense that they have their own separate features and properties and that there are different ones, but we just call that cycling group kind of the regulator protein that are there. So normal cells go through and they display two features, density dependent inhibition and anchorage dependence. So basically when cells divide, they're gonna line up right next to each other in this nice thin sheet. Think skin cells, right? We've got our nice layer of skin cells on the outside here, and then they know to stop dividing when there's, the full set of skin cells. And if one gets sloughed off, or a handful of them, then the cell signals and says, hey, we need to divide, and new ones will form or, push up and replace them. And then, we have our nice smooth layer of cells again, and they don't continue to grow once they know that they are crowded next to other cells. They also display anchorage dependence, so they're going to link and stay in that space. And if anyone knows anything about skin cells, we know we have multiple layers of our skin, so they kind of stay within their location and they don't, just go floating off to different areas of the body. So they're going to be anchored down within their proper space. Now, there are growth factors, again, some additional proteins and nutrients and that sort of thing that are going to be released when cell division needs to occur. So there's all sorts of different types of growth factors out there and experiments that were done to identify these, but this one kind of goes through proteins dependent growth factors that you can always look into, but I think that one's pretty straightforward. The name is growth factor, so they have to be there in order to grow and divide, right? So here's another example of density inhibition. So we have some cells, they're going to grow and replicate and do their division. Something comes through and wipes out a colony of those cells. They'll continue to grow, make that full layer, and then they'll stop once they're completely laid out next to each other because they know they are there. Cancer cells don't follow that regulation of density inhibition and anchorage dependence. So, and they also skip through the checkpoints. So they rush through interphase, which is highly important because the synthesis of genetic material happens during that S phase of interphase, right? And so they rush through that process, and that's where the genetic material gets really messed up. They don't have those cyclin CDK regulators where they say, stop, there's something wrong with your genetic material. Stop, there's something wrong with the growth. They just keep on going and zip through those checkpoints and continue to grow on top of each other. So there's lots of issues that occur in cancer cells, and it's not just like, hey, they all are not properly, they are not all the right size. They might have different amounts of genetic material. They might have multiple nuclei. They may have weird texture to their genetic material too. So normal cells versus cancerous cells. Again, here's our density inhibition. Then they hit that quiescent phase where they just rest, right? They've filled out the entire petri dish. They're good to go. Where cancerous cells will just keep on proliferating or increasing in size and keep on dividing. So we can see that this plate starts to get crowded, and that's where tumors form, where their cells can continue to grow. And then, we might have moles and things like that that represent continued growth of the cells. So I know there were several questions about, like, hey, when cancer forms or whenever there is an error, how many different mutations can occur before we say that it's cancerous cells or that sort of thing. So here's a good example of that. Usually there's um, multiple mutations that are happening with the genetic material or with the cell itself. Um, and then they are able to bypass those checkpoints. And so those mutations hang around, and we are unable to regulate that. And so that's where it goes into the category of cancerous cells. There might be one or two genetic changes that don't affect the cell at all. Again, when we get into genetics, we'll talk about that, but it may not cause the cell to transform. And so they may not be cancerous at that point. But usually it's somewhere around 5 to 7 genetic changes that leads to that transformation process. And then again, they lose their anchorage dependency and their density dependency. So they can then become malignant, and they can start invading other, tissue areas or other organ systems, that sort of thing. That's what I was getting into. So they divide uncontrollably. We'll talk about HeLa cells and why the importance of HeLa cells in biology in class specifically, but basically these are crazy, amazing cells that have been able to hang around since 1951 when the patient Henrietta Lacks was first diagnosed with cancer, and they took some of her cells in order to do some research on them. They never got, this patient confidentiality piece. At the time, she was an African American woman, so there was very few rights protecting her to begin with at this time. And then scientists were able to just pass along her cells from one lab to the next, which is kind of crazy without requiring, a patient signature or the release of her medical information to all sorts of different people. But because of her cells, they were able to identify that they displayed some different characteristics from typical cancerous cells, and they were, for whatever reason, a little or much more hardy, or they were able to survive and continue to divide over and over again all these years. Whereas normal cancer cells, once you put them into a petri dish, they can last, maybe a couple weeks if we have the proper nutrients for them and the proper growth factors being applied to them. But that's the max, where hers are still around. And we know we still have these, and we can still find them in all sorts of different laboratories throughout the world, which is wild. So she had some pretty awesome cells, but again, they were not informing the patient that they had them, and they were being passed along. And then once Henrietta Lacks died, the family members were not aware of this. And so many different treatment plans for all sorts of cancers and other cell-related diseases actually were solved from viewing her cells, whether or not scientists knew it at first or or not. Her cells were huge in identifying some treatment plans. And so that's why we care so much about them, and they're still around today. So, like I said, those cells typically end up dying off in petri dishes, but hers just kind of hung around. Anyway, so that was my little walkabout on HeLa cells, but I'll talk about them more and give you more information on them because they're really interesting. We'll get into transformation later on again when we get into genetics, or if you take biotechnology, we'll do a lab on performing cell transformation and putting in some different traits. But basically, that's when the 5 to 7, genetic mutation limit is reached, and the cell goes through and doesn't display those density inhibition pieces and anchorage dependent pieces. And so then that's where we call them tumors. And then if they don't spread to other areas, right, we can call that a benign tumor, whereas the ones that do, we call malignant tumors. And the malignant tumors perform metastasis, where they can move into different areas of the body or, whatever organism we're talking about. So that could metastasize in other locations. So you might see that word. Again, so if there is an original point of lung cancer happening, and those cells hit the malignant stage, and they're the dangerous types of cancerous cells, that means they can move and they don't display anchorage dependence, and they might go and invade liver cells. And that's where the patient could have multiple forms of cancer within their body systems because those cells were able to move. Or you can start to see them invade vertebrae, and people starting having issues with, are they able to walk properly and stand properly? Laying on the bed might cause pain. Continual movement could even lead to, all sorts of organ systems. So that's the dangerous part. We should probably already be aware of that there are different causes to cancer. I have all sorts of stories and connections with my family about carcinogenic, I can't say it, but about carcinogens and how they have impacted them, and whether they were, in direct contact, or it was, through their family members who were smokers or whatever. There are some big risk factors that we can go into and just make sure you're being healthy and you're aware of what your body can handle and what is normal for your body. And you might even be able to detect when things are kind of going badly, and perhaps adjust some of these risk factors early enough so that cancer doesn't form. But here's a whole list of some potential ones. Our bodies defend against cancer in multiple ways besides our cyclins and CDKs, and then beyond our checkpoints that we have. We all have these genes called P53 genes. And in cancerous cells, in about 50% of the cases, there is a mutation with the P53 gene for our cancer cells. And so there's some sort of issue going on where these genes no longer can fight off cancer. So they're called the guardian of the genome because they're actively functioning, and they realize that there's something wrong with our genetic material, and they can go through and fix it and repair it, or get some protein factors to go through and repair it. And if not, the P53 gene initiates cell death, which is also known as apoptosis, if it can't be repaired at all. So good example of that is being exposed to the sun so long for a certain period of time. UV rays are damaging our skin cells. P53 can easily kind of go through and slough off and kill off those cells through apoptosis and keep the good ones. So I'm going to just zip through here. There are other examples that go along with genes that function and malfunction that are associated to cancer. But this last piece is on apoptosis and describing what that is. And that is, again, controlled programmed cell death. So, a cell is going to reach its maximum amount of replications or cell division processes before the DNA starts to show damage and genes start getting exposed to various factors, which can be very dangerous for the cell. So, once they've reached their maximum cell divisions, they'll hit that point, and apoptosis will naturally happen, where like our lysosomes are going to be very active and enzymes will come through and start breaking apart the cell. It'll shrivel up and die off, so that if there are any mutations with that cell, they're not passed along to the next cell division. And so apoptosis can happen naturally with hitting its maximum cell division points, or it can happen through that process of our cell cycle when it hits that checkpoint or go no go phase. Um, and it's an irreparable damage type thing that's happening, apoptosis will be signaled. Or our P53 gene, our guardian of the genome, will come on through and say, hey, I can't fix this. This is really dangerous. Genetic material is out of control. Apoptosis.